Hi everyone, good morning. Thanks for joining this live. Today's lecture is gonna be a bit of gonna be quite long, but please bear with me. Today we are going to discuss host microbe interactions, and the objectives for today is as follows. We will name the site where normal biota is found in humans and summarize the type of normal biota found at each site. We also name contributing factors to how the microbes form in infants. We explain the importance of the microbiome. Sorry, I meant microbiome, not microbe. So we will explain the importance of microbiome. We will list Koch's postulate and state the overall goal of Koch's postulate. We will define the following terms: true pathogen, opportunistic pathogen, virulence factor, and infectious dose. We also identify the major type of infections. We describe the 5 stages of infection, including major steps or mechanisms for each, and using key terms. We define the periods and label a curve representing the course of clinical infection. We will also distinguish between direct and indirect transmission of disease using key terms for the nodes of transmission. And then we will compare and contrast the various type of reservation sources and carriers, providing examples for each. We will then list and define the 4 types of epidemiology cases and there are 3 major types of epidemics, and then we will define the terms incidence and prevalence and calculate each. So this will be the outline for today. First, talk about the microbiome, and we will also talk about the progression of infection, specifically pathogenesis, and then thirdly we talk about infections with population as it has to do with transmission and epidemiology. So first, let's focus on the microbiome. So microbiome is basically the total collection of population of all microbes in your body, basically the microbes that have colonized the body. And they are also known to be called several names such as resident microbiota, or normal biota or flora. They include all range of microorganisms, including bacteria, fungi, protozoa, and viruses. They differ based on the location that they can be found in the body. Also, they can differ due to age or genetics or maybe health conditions. Um, Places in the body that is known to not have microbiota present is mostly the bloodstream and then the brain. And as you can clearly see, if you take only the mouth alone, you realize it contains a lot, like streptococcus, candida albicans, and a lot of microbes. And the same thing goes for your GI tract, in your intestines, you have a lot, and then even in your um mucosa linings in the lung airways, you also have some present too. So then you really can tell how diverse microbiota is in the whole body. So now let's talk much more about the sites where we can find normal microbiota present. So first one we talk about is the skin and adjacent mucous membrane, including the external ear and eye. And clearly you can see that there's variety of microbiota present. So if you take with just the skin alone, you can clearly find them all over the place. For example, just the toe web, you can also find some of microbiota present. And then this microbiota that falls under this category mostly are gram-positive, especially you have Staphylococcus species, and they are mostly present on your skin and on the surface of the body. The second place you can also find microbiota is through your oral cavity, mouth, and throat, and this is mostly also Staphylococcus species. And then it can vary. The population can vary from person to person depending on their hygiene and also a lot of different factors like genetics or maybe if they have specific diseases. And then in your oral cavity, you mostly have plaques, which is basically important microbiota forming biofilms on your teeth. So it depends on how frequent you clean your teeth. Maybe you might not have a lot of plaques on it. The next category is your GI tract, which is the most common one that we all know. They are the majority of them are mostly E. coli, and most of them are gram-negative bacteria. What makes them more distinct from the ones we've talked about so far is that they are predominantly anaerobics, and some of them are also aerotolerant or facultative microbes because down there in your GI tract, you don't have much of oxygen flowing, so they have to kind of be resistant. They have to be able to grow without it. Um, because this is the well-known place to have microbes, there's a lot of studies that have been done relating to your gut microbiome infections interactions. And recently, they've also been studies showcasing the buildup of the initial infant buildup of gut microbe. Once a child is born, once their gut microbe are building, it have a relationship with the brain's function and forming. Basically, a relationship between gut microbiome, baby infant's gut microbiome, and brain cognitive. That shows you how diverse and important having this microbiome in our gut is. It is very extremely important. So the fourth one is your respiratory tract. Previously, it was thought to be the most sterilized place in the body until recently they observed some bacteria also present in your upper airways. There's actually a lot of them, as you can see in this figure, Acinetobacter, and the list goes on and on. So bacteria, they actually lower respiratory tract space, and they are not too much present in your respiratory tract. There's just low abundance compared to somewhere in the mouth where you have a lot. And about 30% of them carry Staphylococcus aureus. About 30% of us carry Staphylococcus aureus in the nasal cavity. And as you can see from most of our lab, we ask you to kind of take a nasal swab, and so that um, we can grow them on the blood again and see what kind of microbes we get. And then this is mostly because we have microbes present in both our mouth and the nasal and almost everywhere. And and as I pointed out, we in our lab we did both oral and nasal swab, and as you can see that um, most some of the microbes overlap between throat and oral cavity. So if you did both, you realize that from your experiment you might see similar microbes from both places. So the next place to talk about should be the genitorial urinary tract. So the urinary tract is sterilized except for some short portion of the anterior urinary tract where lactobacillus present. So in female reproductive system, there are more diverse bacteria. The most common one is Candida albicans, and then we have lactobacillus present, and um, and their whole job is to try and keep the population of Candida albicans down, um, so that to balance the microbiome down there. And then in the male reproductive tract, it's mostly sterile except the tip of the urethra, which will also have both the lactobacillus and also streptococcus present. So when does the microbiome start to grow? So the first one that we talk about is in utero. So basically, it happens during pregnancy. So it's believed that the womb probably have its own microbiota, and then it's supposed to be there to strengthen the immune system of the kid and also protect the kid. And then during birth, vaginal canal and C-section delivery also contribute to initial microbiomes to the baby. And also when the baby is breastfeed, specifically through breast milk, the mom passes some IgG and also some microbiome or some microbes also to the kid to help build their immune system. And also through caregivers and our siblings giving string kisses and skin touch with the kid, they will also transfer some microbe. And also through environment, when the kids start crawling, picking up stuff, putting stuff in their mouth, this whole process help build up their microbiome. So what are the importance of microbiome? So the microbiome is actually important in several ways. One of it is important for food digestion, especially the ones in your GI gut and then GI tract. And it's important for food digestion. It's also important for synthesis of vitamins and amino acid. They also influence development of your organs, and they also antagonize against pathogens. So when you get infected with pathogens, most of your native microbiomes, they are the first to attack the microbes to keep their populations down, and they also help in the build-up of your immune system. So you do realize that just a single bacteria can give you a lot of importance in your microbiome. This single bacteria, when you think or you pick a single microbiome, it gives you a lot of importance or a lot of functions which contribute to your well-being. Also, as much as they have good importance for our well-being, there have been also some preliminary studies which have been associated, in which we've associated them with some risks involving certain diseases and mm, certain lifestyle changes. One of these, some of the examples include the Crohn's disease, obesity, heart disease, asthma, autism, and diabetes. And this basically happened due to dysbiosis, basically imbalance in the microbiome, which will then be associated with diseases. And then, for example, if you have, as I pointed earlier, the gut-brain axis. So if you have an imbalance between the gut-brain axis, especially as a child, you might have neurodegenerative disorders, autism, or pneumonia, or psychiatric disorders, and you just keep going on and on and on. So, in order to fully understand the importance of the microbiome, research have to first isolate the pathogens from patients and then diagnose them. Isolating pathogens from patient can require distinguishing are required so that you can distinguish between pathogen species from the normal biota in cases where you have to distinguish between the foreign biota and the normal biota. That's when you are trying to understand which pathogen causes a specific disease. And in order to do this, you have to sample the microbiota from a specific given location. So, because there's a lot of them all over the place. For example, if you want to sample gut microbiota, you only have to select or sample exactly from the gut. Then you can go ahead and do further research to distinguish between the normal gut microbiota and the foreign one that is not normal. And something that I want to point out is that some of these gut microbiota, the normal biota are opportunistic pathogens. Basically, um, they are more present and more active in an immunocompromised situation. They don't actually cause the immune compromised situation, but once your immune system is compromised, they take that opportunity to cause more harm. And also, some of them might move from where they actually originated and go and reside in abnormal places where they are not supposed to be and cause more problems. So try and practice this. We also discuss this in class. So now that we fully understand what normal gut microbiota is and what opportunistic, sorry, now that we've understand what normal microbiota is and what opportunistic microbiota is, let's talk about their progression of infection, specifically pathogenesis. So just imagine this as you, which is your body. Um, you are just basically colonized with like trillions of resident microbes in your body, and then they are playing a role. They are just playing a role in protecting your body. But what happened is that both the potential pathogens and then the transient pathogens, which are basic, which are basically colonized in your body, will end up being exiting your body, or they'll just exit your body and be gone. In some cases, both the potential pathogens and occasionally some resident microbiota as I point out, the opportunistic microbiota can cause infections, and then your body can eventually withstand it. You get treated, and then those who cause the infection will be cleared out. Occasionally, those who cause infection do lead to a chronic infection, which results in disease. And if you are unlucky, some might lead to death. In some cases, you get a disease, you get treated, the microbes will be eliminated again. But if you are not lucky, you get a disease and you get treated, some of the microbes will be gone and some will still be left and then will still come back to your body. You are the holobiont. Basically, uh, um, you are basically a container that contains a lot of microbes. So they will come back to you, and then the cycle goes on and on and on. So it does tell us the relationship among us and the microbes. So now let's understand Kush postulate. And the overall goal of this postulate, or the overall goal, is to determine the etiological agent of an infection. Because we have an infection, we have to know what exactly is causing the infection. And to do this, you have to first point to the causative agent, basically the agent that caused the disease. And Robert Koch in the uh, um, early 800s developed a standard for determining what is a causative agent for a specific disease. And what he did was to determine causative agent for anthrax. Basically, he did a series of proof that established a classic criteria for etiological studies. So this is what we now took back from the early 80s. Scientists took over, and it has been useful in identifying causative agents of certain disease. So now let's talk about the postulate and how it works. So basically, what he did is he first found evidence. What he did, the whole postulate is being outlined as: first, you have to find evidence of a particular microbe in every case of disease, but not that from a healthy individual. So first, let's say I'm giving an example. Let's say you have a person die from maybe a gut microbiota infection. You sample from gut microbiota, the whole gut microbiota of the disease, and culture, and then you take one of a healthy one. So now let's move to the step two. You isolate micro from the infected subject and create a pure culture in the laboratory. So now that we've have these microbes grown from both subjects, we will take microbes from the disease and then try to create pure culture, basically. And then the third part is then you try and inoculate a susceptible, healthy patient with some of the isolated microbes that we've already done here. So we take some of the microbe and inject it with a healthy animal. If the healthy animal um, die from the re-isolate we took from the disease, then that might be the causative agent of that disease. If it doesn't die, then that microbe is not. But there are some complications or caveat to this postulate. Um, you can't culture all of microbes, right? So some of them are not cannot be cultured on this artificial media, and some of them will need more of a specific pathogen, it will need a specific host in order to grow. One example is like HIV and viruses. Viruses need like hosts to grow in it, and then it's super unethical to culture virus in humans. It's super unethical. And one side effect is that some diseases are caused by multiple agents, so it's very difficult to find out which particular one is doing it unless both pathogens are, or more than. For example, if it's a disease is caused by more than one pathogen, you need both pathogen present to be able to cause the disease. One of such diseases is pneumonia, where you need a variety of microbes to cause that disease. So if you are doing a single isolate of pure culture injections from the Koch postulate, you might not be able to find the raw causative agents. And lastly, is that you end up culturing the opportunistic pathogens. As we talk about the opportunistic pathogens, some of them will move to a different part of the body because that part of the body have already been infected and is weakened. So the opportunistic pathogens will move there. So if you culture microbes from there, you end up culturing the opportunistic pathogens, which are not the real causative agents. So then it brings a problem. of how do we then categorize the pathogens? What are true pathogens and what are opportunistic pathogens? So the true pathogens are the ones that are actually causing the disease in the healthy patient, resulting in immunocompetent persons. And then the opportunistic pathogens are normally um, not typically infectious to the immunocompetent person, but cause diseases when the host immune systems are compromised. So once the true pathogens have caused the problem and the immune system is compromised, then the opportunistic pathogens will take over. And then sometimes the second part, when they also become established in part of the body that is not natural to them. So now let's talk more about opportunistic pathogens. The first one we talk about is the host defense that are compromised. So what opportunistic pathogens are found in places. So so we are talking about the mechanisms in which we can find opportunistic pathogens being present. So one of the first mechanism is when a host defense are compromised. So the host defense might be compromised through a lot of series. So first one can be primary immunodeficiency. Some can be secondary immunodeficiency. It can also happen when you are old or when you are super young or when someone have cancer and they have to undergo chemotherapy, or maybe someone is sick or they need blood transfusion, so they have to first suppress the immune system. And it even happens when you are pregnant, the mother's immune system goes down so that it doesn't attack the infant. And then sometimes when you get under physical stress, like and mental stress, there's a lot of ways in which the host body can be immunocompromised, and then the opportunistic pathogens can take advantage of the situation. The second situation is when they become established in part of a body that is not natural to them. So, for an example, is E. coli. Naturally, E. coli should be resident in your intestines and particularly part of your rectum or in your rectum because that's where your feces go out to. So, let's say in some instances, you have E. coli present in your uterus and then um, in your sexual organs and also in your ureter. These are not president or these are not the resident place where a microbe such as E. coli should be. So mostly the body tries to take care of it. Uh, um, through flashing out or peeing. So if it's resident in places like the bladder, you end up peeing them and just getting rid of the E. coli present there. So this is just a clear example of E. coli, which is supposed to be in the GI gut. And and when we talk about your lower body in your rectum, but it's being found, E. coli is now being found in the different place where it's not supposed to be. If you don't get rid of it, you're gonna end up having infections. So um categorize pathogens, we have to first understand how potent they are. Basically, we are talking about virulence. So virulence basically is a related capacity of microbes to establish itself to the host and cause damage. And in order to understand the virulence of a pathogen, you first have to understand or calculate what their virulence factor is. So virulence factor is any characteristics or structure of the microbe that contribute to it establishing itself. So the pathogen will need to have a virulence factor in order to be able to be more potent and cause disease. Some of the virulence factors include toxin production, and they also will acquire some special nutrient to invade. They can also include induction of injuries to the host response. So violence factor contains a lot of characteristics that the microbe need in order to establish damage to the host. And one thing we also want to know when we want to characterize a pathogen is to also understand um, their infectious dose. Basically, infectious dose is the minimum number of microbes required for infection. So we also have to know how much microbe is needed to cause an infection. So for example, just focus here. So if you have a lower infectious dose, it means, let's say you need just two microbes to cause an infection. It means they have a very high valence rate compared to when you need a lot of microbes to cause an infection. It means they have a lower valence rate. So that's how the infectious dose work. An example is here. So for example, you need um 1 to 10 Francisella tularensis to cause the tularemia disease, right? Compared to here, you need 1 million salmonella to able to cause salmonellosis. So you understand, like, this is more virulent compared to salmonella. So that's how infectious dose is being calculated, and that's how it works. So now that we understand all of that, we also have to talk about types of infections. So infection can be localized. Basically, they are just localized within a specific tissue, and mostly we see them when you have like skin diseases, like fungal disease on your skin and warts, and it's just present on one particular area. The second category is systemic. Basically, it's just spread throughout all of your tissue. Mostly, it's been done through your bloodstream or your fluid tissue, and it's just everywhere. And then we have focal. Focal basically means the microbe will break loose from a local infection and then is carried to other tissue. One example is the streptococcal pharyngitis, where it starts from there and then turns into a scarlet fever disease. Another category or another type is polymicrobia, or basically a mess, where you need more than one microbe to contribute to the disease. One example is seen is mostly found in dental caries, where you need more than one microbe to basically cause a tooth decay or some or damage to your tooth. And then we also have the primary and secondary situation. This is where the opportunistic pathogen comes in, where you have a primary infection, a primary pathogen causing infection, and then you have a secondary, which is the opportunistic pathogen coming in and making it worse. So let's practice this, and we also talk much more about it in class. So now, what are the stages of infection for a pathogen? We have 5 stages. The first one is to trying to find a point of entry. It can be your GI tract, can be your skin, urinary tract, any place where the microbe can find and get into your body. Once it gets into your body, you have to attach firmly to the host cell that it plans or tissue that it plans to attack. And it's going to do this using the fimbriae of the microbe or viral spike or surface proteins. And then once it's established, affirms or attaches firmly, it have to try and evade your host defenses, because in the last few classes we talk about your host defenses attacking the microbes or the pathogens when they get into your body. So the pathogen have to first try and also avoid your host defense through all kinds of ways, including avoiding phagocytosis and also absence of specific immunity, trying to invade your whole body. And then after it has invaded your host defense system, it have to then go ahead and cause damage or cause the disease. They do this through releasing of series of enzymes or toxins, or also inducing an excessive host immune response, sometimes causing autoimmune diseases and things that have to come to your body attacking itself. And then lastly, it have to then exist the host. You can do this through your respiratory tract, where you have some people who just sneeze and just give off the disease out, or skin cells, or fecal matter, or blood. We'll talk deeply about each single one of them. First, let's talk about the first one, which is the point, the finding an entry portal into your body. So entry portal is basically the route taken by the microbe to initiate your infection. To initiate an infection can be from skin, your nasal, oral, it can be everywhere. And then what you have to realize is that your endogenous microbes can also lead to super infection when you have other microbes present. Basically, you're just taking opportunity of this infection. And that's the opportunistic microbe we've been talking about. So now that they've gotten into your body, let's talk about the next stage, which is attaching to a host cell. And They mostly do this through film drive. For example, here where you have a bacteria coming, it uses a film drive to attach to a host cell. They also come in through capsules where you have a bacteria cell coming in and it's just coming in through a capsule. And for example, in a virus, they also come in using their spikes to try and attach to the surface of the cell. And then for example, in some bacteria like this one, they have to form a biofilm, try and protect themselves basically from any other attack that will come to them. So now, as I pointed out earlier with biofilm, they have to form biofilm and then it helps them stay attached and it helps them protect, it helps protect them too. So these are example images of biofilm. This is Staphylococcus aureus having a biofilm, Staphylococcus aureus biofilm on a suture that on a suture. And then here, this is E. coli biofilm on a bladder epithelial cell, and it's present here. And then the most common one we all know is a polymicrobial biofilm plaques on your teeth. It's more present if you have a little bit of I mean, everybody have it, but as to how extreme you have it depend on your oral hygiene. So the third stage is to evade the host immune system. So evasion basically is the mechanism where the pathogen will use to detect or prevent or limit the distraction of the host immune system against them. So this is where we consider the virulence factors coming in. So they may differ within a microbial group and species. Every microbe or every microbial species have a way it evade the immune system. It's not like all of them have the same thing, but basically all the mechanism they are using is what we classify as the virulence factors, basically the factors that help them survive. Now let's talk about it one after the other. The first one we talk about it is to evade phagocytosis. An example is that you have an encapsulated bacteria such as Streptococcus pneumoniae, which just marks its all palms present on its surface, and then it puts it under the capsule, and then it just the phagocytotic cell cannot recognize the palms and then engulf it and then degrade it. It's just cannot do it. And we also have some situation specifically in Staphylococcus aureus where it just goes ahead and then it kills the phagocyte directly by producing leukocidins targeting the phagocytotic cells and just killing them instead of the phagocytotic cell killing the Staphylococcus. They kill them rather. And then if they are not in some microbes, if they are not able to evade the phagocytotic cell and then they just get engulfed. Then they tried also evade the mechanisms within the phagocytotic cell. So one example is to avoid phagolysosome killing. So the first one is number one, they escape phagolysosome. So they can change the pH of the phagolysosome or change everything where they cannot be engulfed. So they just escapes and go away. The second one is to prevent phagolysosome fusion. So phagolysosome fusion, they try and make sure that they are not fused with the phagolysosome. So kind of um you cannot fuse with the phagolysosome, it just escapes and prevents it from fusing and then degradation. And the last one is the third one where they survive the phagolysosome. So even though lysosome are secreted, in the microbe is basically engulfed, and then the enzymes phagolysosome are secreted. It just basically escapes the degradation of the enzyme. It's just resistant to it, and it just eventually get away. Some of these microbes or bacteria include Mycobacterium tuberculosis, which causes tuberculosis, and then Listeria monocytogenes. And they live inside the phagocytosome and they just divert the killing mechanism. The third one is to avoid recognition by antibodies and then lymphocytes. So we have some bacteria which will produce enzymes towards some of these antibodies. One example is they can secrete proteases that degrades, let's say IgA, it just breaks it down in the mucosal surface, and then IgA never targets the bacteria. An example is um, antigenic variation, where they also alter the surface of the surface antigen to minimize the detection by the antigen. So, like we told you, it's like a puzzle. So they change the surface of the puzzle where the antigen cannot fuse to the receptor again. So it doesn't recognize it, then it cannot bind to it. And some of them also have the extremeness. An example is Streptococcus pyogenes, where it also produces antigens or self molecules. It produces molecules that mimic the self antigen of the human cell, so that T cells cannot recognize it and take care of it, or the cytotoxic T cell cannot recognize and take care of it, or the natural killer cells cannot recognize it because it have some of them will mimic the protein MHC1 that they will present it on the surface, and then it look like self cells. Basically, they are not self cells. So one way they can do that is to use hyaluronic acid, which is a polysaccharide present in a human tissue, and they just present it as a self antibody or a self antigen. Sorry. The fourth one is that they hide out in other cells to avoid extracellular detection. One example is Shigella. Basically, you have a Shigella, and then it basically invades epithelial cells by spreading from cell to cell through actin filament. So you have it here. It enters the epithelial cell and then invades through actin filament, moving from one cell to another cell. The fifth one is that they regulate the the infected host cell death. So the first mechanism is that they prevent the host cell death to prolong their life cycle. So they are making sure the host cell doesn't die. They just make sure the cell is alive so that they can also prolong their life cycle. Because if the host cell dies, they also die. The second one is to block MHC1 presentation and then present a fake MHC1 and fake natural killer cells. That's the one I had talked about earlier on. So it goes in there. They then prevent a host one, and then sorry, they prevent the host cells from making MHC1, and the pathogen will now produce a fake MHC1. So the NK cell sees it, they think, oh, this isn't self cell, and then they ignore it. Basically, they are not, it's just a trick. So look through this, we also practice this in class. So now let's talk about the fourth stage, which is causing disease. So causing disease, we also have to we've already talked about the violence factors. We also will talk about the range of the mechanisms or direct or indirect consequences of the infection. So we also have to know um whether the pathogen is causing a direct consequence or indirect consequence from the infection. So there are three major mechanisms mechanisms that falls under this category of causing diseases. The first one is secretion of exotoxins and exoenzymes. So basically, now the bacteria is inside the body. How does it cause the disease? So it can do it through the secretion of enzymes and toxins. Secondly, is to also modify the host DNA to disrupt the host cell function. And thirdly, they can cause they can also cause disease through causing the host excessive or inappropriate micro response to the microbes. That is more of an indirect form. The first two are both direct form, where they are just doing it themselves. They are just changing the host. They are basically affecting or causing the disease by themselves through maybe secreting enzyme or disrupting the cell function and everything. The second one is just to cause the host cells to do the damage instead of they themselves. And then all of these are often seen in your body externally as a manifestation of the disease symptoms. So it can be so it can be direct and indirect type of infection that they will cause. So now let's talk of the first one, which is secretion of exotoxins and exoenzymes. So the exoenzyme basically they will break down and damage the tissue in order to dissolve the host defense barriers and then promote the spread of the microbes into deep tissue. So as you can see, the bacteria is here, secrete enzyme and just degrade the barriers which are needed to so in your host cells, in your cells, you need these barriers to prevent the entry of anything into your tissues or into your body. And then, for example, if this is the bloodstream, you are just protecting yourself from getting anything within the bloodstream. And then you have the bacteria basically degrading everything and making sure they themselves get to enter the body, get to enter the bloodstream or the specific tissue in which they are. So these epithelial cells are basically linings of specific tissues. And they protect your organs from being infected. And then you have the bacteria going in there and degrading them. And then we have the second mechanism is exotoxins, which are released by the microbes and they directly damage and kill the cells or impact the cellular function. And then we also see this as symptoms in the host. So instead of degrading the damaging the tissue or breaking the bonds between cells, as you can see here, this time around they attack the cells itself and just kill the cells. One of such examples is what we've seen so far in the lab experiments where we use the Staphylococcus species. We have the alpha hemolysis, beta hemolysis, where the Staphylococcus is just attacking the blood cells. So, in relation to the release of toxins, bacteria can also cause a third one, which we call as botulinum toxin. So, an example is the Clostridium botulinum toxins, which basically causes botulinum. What the toxin does is that it goes and just block the transmission of nerve signals to the muscles, and then we prevent basically the release of acetylcholine. So here is your muscle cell, then you have your neurons present. The neurons have to secrete acetylcholine to the muscle cells and then instruct your muscles to be able to move, right? Trigger muscle movement. And then you have the bacteria release a toxin. And then the toxin goes in there and just block the release. And then it will result in you having a flaccid paralysis. So this shows how diverse the toxins can work. So they can either block the transmission of signals or cause a disruption of the cell itself or cause the disruption of barrier of tissues. So now let's talk about the second one, which is modify host DNA to disrupt the cell function. So they can go in there after destruction, go in there and change the genetic expression or the gene expression of some host cells, which basically they are causing epigenetic changes in your host cell. And they do this in different mechanisms. It can be through DNA methylation or histone modification, or they can go in there and then change the mechanism of your body to produce only microbial proteins and enzymes. So they can also cause the third one is that they can also cause the host body to respond excessively or inappropriately due to their microbes doing that, and we've talked about it as the indirect form. So one example is that they can cause damage by inflammation and basically trigger cytokine storm. So where a lot of cytokines are being released, and then you have like a huge inflammatory response coming from your body. And then also sometimes they can do trigger the this through acquired immunity. So once you get infected, the your body have to respond. And this example comes from here. Now we have B cells releasing antibodies. Antibody the bacteria sees the antibody coming and then it start producing mimicry of the molecule. We start generating mimicry of the antibody. Once the antibody is the mimicry is being done, the body now release more of it and basically mimicking the acquired immune system and then causing a lot of diseases basically through overreactive um, antibodies being released. So one example is here. Then the bacteria is having the interaction with the antibodies. then it starts producing the same antibodies, which is then kind of irritating the heart. So the antibodies goes into the bloodstream, goes inside the heart, and it's just irritating it, causing rheumatic fever. And as we can see, it's just super bad, basically. It's just super bad. So those are the different ways in which the bacteria can cause inappropriate immune response. So inflammation or through acquired immunity. So now it has done all the damage it needs to be done. The bacteria have to exit the body because the end goal of every pathogen or every or bacteria or every pathogen is that once the damage is done, it have to leave the body. And basically, it can leave the body through a lot of ways. One of them is through your airways when you sneeze. It can also come from your skin cells through opening and lesions. You have to pee them out. You also can do it through feces. They can come out through removal of blood during blood transfusion. It can be done through removal of blood when you have a your blood being drawn out. And then in some rare cases, you can have insect bite where mosquito will just bite you, suck your blood, and just um move it to the next person. Please practice this. We also practice this in class. So now let's talk about the course of clinical infection or the whole process of clinical infection. The first one is the incubation period, basically, where you have your initial exposure to the microbe. It will be lasting around 2 to 30 days once you come in contact with the microbe. And then the next phase is the prodromal period. Basically, your earliest notable symptoms appear. So this is mostly when the bacteria have gone in, attaches itself to your host body and has started causing damages to the host cell or inducing immune responses. So that's when you get your symptoms. Then it moves to the height of infection or the period of invasion where the pathogen is doing multiplies. The pathogen basically multiplies at high level and is causing great violence into your body. So that's where their greatest violence is at. It's just at the peak. It's just causing more infections compared to here where you have a little bit of symptoms showing. Here you have the greatest of symptoms showing, and the bacteria just attacking your body. And then it moved to the third, one, two, three, four, fourth stage, which is convalescence period, basically where your patients will respond to the infection and symptoms decline. So here, your host cells start responding your host body start as responding excessively to the infection that is being caused. If you are lucky, the symptoms have to decline because your host cells have been taken care of or you took drugs and you've been able to overcome the disease. If you are not lucky, then I guess it's leading to your death. And then the fifth one is the continuation period, where only in some infections, as I was describing, some infections, you have some of the microbes lingering around and causing subsequent infection. So now let's talk about the last part, infections with populations. And that's we'll talk about transmissions and then epidemiology. So now you have disease. We've only been talking so far about disease in one specific host, but how does it transmit from one host to the other? One of the ways it transmits and the common ways is the direct transmission. Basically, you need a direct contact or close proximity with another person. The most common one, which we saw so much in COVID, is coughing and sneezing. And then we also have places where you can get it through, you can share it through your food, saliva, kissing, and sexual intercourse through STIs, through touching. These are the most common ways to have a direct infection. Mostly happens through direct contact. So, in view of this, who has recently reported a redefining respiratory particle transmission? So, at first, we thought you have to be super close to get the respiratory kind of infection or transmission. But until COVID came and we realized that even someone called from extremely far distance, you can still get it. So this diagram is just showing that some bacteria have airborne transmission and which can easily be done from any distance through the air. And then effectively someone have to do an inhalation and just get it. And it just most common one of them is the one coming from your respiratory tract, and that's what it can be done. And then the second one or the second category is through direct deposit, which is normally a short one. And then it's most also through the air. It's just semi-ballistic trajectory compared to the one like COVID, where you have to cough and it's easy to get. And then you have to come a bit closer. This one, you have to come a bit closer to the person, where you have to get a direct deposition in your mucosa, mostly through your mouth, nose, and eyes. And this is more common when you are, I say, you kiss somebody, and then this is more common way of transmission. So now that we've understood this, let's talk about the indirect form of transmission. So pathogens can be passed from infected host to infected host to another person through an intermediate conveyor. So basically, the intermediate conveyor can be fomite or fomite, basically an inanimate object such as doorknobs, faucet. If people have bacteria on their hand and they keep touching it, everyone will have it. So imagine you're in the lab and then you touch all the handles with you touch the bacteria and touch all the door handles with it, you'll be able to get it. And you realize one of the experiment we did was for you guys to go out and sample places like doorknobs or any on even on the bench to see if we can culture microbes. That's the very first thing we did in the lab, and that tells you that that is an indirect disease transmission. And then the other one is parental, which is basically injections to the deep tissue. It can be done through knife, broken glasses, and needles. And then we also have when it's being transmitted through vehicle, basically non-living natural material that can transmit pathogens. So common one is food. If a food is infected with a pathogen, let's say bacteria or microbe, you can easily get it. Common one is soil. That's why you guys went out there to culture the soil in the first few weeks. It's actually the first lab we did for you guys to sample from the soil. We can also have some coming from water. We can have some coming from airborne, which is basically dust or moisture particles in the air. And then the last part that we will talk about is more fecal oral. So when some pathogens are being, most pathogens, some pathogens are being found in fecal matter, and then when someone defecate or something, they just defecate or have feces around places, people can have it. So they can have, let's say someone have feces on the format like a doorknob, or they have feces in food or soil, and you some way can land into your body. One of these is E. coli. E. coli is supposed to be only delivered through feces. So once you have an E. coli infection, it means you came in contact with feces either through formal, vehicle, or parental form of transmission. So, to understand disease transmission, we first have to understand the source. So, the source basically means the individual of this from which the infection is acquired. It can be a person, can be formal, it can be direct, and it can be indirect. And then we have to also know what the reservoir is. So, reservoir basically means a primary habitat from which the pathogen originates. So, basically, someone, a person can be the reservoir. So that's from which they are the main source from which the disease is being disseminated. And it can be living and non-living. So now let's first talk about the non-living reservoirs that we know. One example is soil. It can be water, can include air or buildings. And they mostly are saprobes. They are found everywhere. Bacteria or pathogens that form under this category are saprobes. They are found everywhere. They cause little harm to humans. And we, in some rare cases, we will have some opportunistic pathogens. We can have some opportunistic microbes and some pathogens doing the same thing. And their mode of infection is the most common one through a vehicle. So it can be through soil, it can be through anywhere, doorknobs, water. Now let's talk about the living reservoir. The most common one is humans, as I pointed earlier on, and basically someone who is sick and is a carrier and is just transmitting it. And then there are types of carriers. falling under humans. So we can have asymptomatic carriers. Basically, they are infected, but they do not have the symptoms. So one example is COVID-19. Someone might have it, you don't know, or someone might have an STI and you won't see it until you are given the STI. And then the second one is incubating carriers. They are infected and they are just within the incubation period, so they are not showing any symptom until they give it to somebody. And the third one is co covalescent carriers. Basically, they are infected. They are within the convalescent period. They show no symptoms. One example is hepatitis A. You wouldn't see it until you go and kiss somebody and you get transmitted with it, or you have a contact with the person, you just get it. And then another one is the chronic carrier. So these people actually have latent infection. They don't show symptoms. An example is tuberculosis and so typhoid fever. They just have latent infection here and there. And then the fifth one is passive carriers. They are not infected. They are only more of contaminated. So one example is the healthcare associated infections. This is just passive carriers and they are just um spreading it around. So not only humans can be living reservoirs. They are the most common one. Another common one is arthropods of vectors. So arthropods or insects, arthropods or insects. So basically, they can come from mosquito bites, ticks. They can suck your blood and then move it to the next person. And then the third category is also other animals or other vectors. And all of what we've been talking about is that the living reservoirs can be spread through both biological and mechanical vectors, right? So the biological vectors are first infected. They can get the infection and they can send it to the next person. The mechanical vectors, they are not infected. They just carry the pathogens on their body and just send it around. For example, you can have a fly which will carry the pathogen, or a roach which will have the pathogen and just send it around, compared to a chicken or a bat who might, in our case, might be infected in a bat, which in our case might be infected with COVID-19 and might send it around, or a chicken which will have like a chicken pox or something and just send it around. So, in relation to biological vectors, since they can get infected and send it around, they can include animals and humans, then this leads us to zoonosis. So, zoonosis is basically an infection indigenous to animals but are also transmittable to humans. So, human is the dead end host, but they have to be get it through an animal. And this is actually spread mostly through human to animal contacts or human to animal association. So if you have a car, if you have a pet, or you are eating meat coming from human animals, mostly if it's uncooked, you might end up getting zoonotic diseases. So let's practice this. We talk more. So now let's talk about epidemiology. It's basically the studies of disease frequency and distribution within communities. And in able to study epidemiology of a specific disease, you have to first collect and analyze the data on the disease. You have to design strategies to prevent the spread of the disease, and then you have to also report um, um, or notify the report you've already done about the disease. Mostly, you require report it to the authorities, whether CDC or FDA, or depending on which country you are, you have to report to the authorities. Now, what are the type of epidemiology cases? The first one is a endemic, which is basically a steady frequency over a long period of time. So you have a specific disease just studying it through a long period of time. And then we have the sporadic cases where you have occasional random locations, irregular intervals. So let's say the one will be North America, another one will be South America, just randomly. Then we have epidemic, basically when the prevalence is increased beyond expectation and it's more like localized within a specific place and it's just going all the way up. The last category is pandemic, where you have epidemic spread all across continents. One example is COVID-19, which we all experienced. So, in order to fully understand epidemics, you also have to first understand things like point source of epidemic. So, you have to know where the source of the epidemics happen. One point is one thing we have to know is the point source, which is basically if sorry. Sorry. So to understand epidemics, it's mostly caused from one single source, which is a point source epidemic. So this is a type of epidemic. So the point source epidemic is a type of epidemic, and basically point source by its name means, let's say all of us went to like a party, we went to grab a crab cake, and then the crab cake is infected, all of us will get it. That means we all get it from the same point of contact or same points, same source. That's what we term as the point source epidemic. Basically, epidemic arising from the same source, multiple people getting it from the same source at the same time. Then we have the common source epidemic. Basically, it's just common exposure to a single source of infection over a period of time. Let's say the ice machine in the lab, someone smears E. coli on it, and as time goes on, I touch it, you touch it, your colleague touch it, we all keep touching it throughout the whole month. By the end of the month, we all have E. coli infection. We all got it from the same common source, but not at the same time. Compared to how that, so the same time is point source epidemic, but not at the same time is common source epidemic, and it's more common in like you can get it through like contaminated water or plant infection, food product, equipment, and restaurant worker can also be a carrier of hepatitis A, and through poor hygiene practices can spread it. So one is simple example is like imagine the ice machine in the lab, someone spread E. coli on it, we all keep touching it throughout the whole lab or throughout the whole month, and we all get it. Then we also have a propagated epidemic. Basically, the pathogen is spread from one person to person. One common one is COVID-19. One person have it, he sneezes on these two people have it, they keep spreading it and keep going. Propagated epidemic is more like one of the common ways of spreading disease. When someone have it, they can give it out easily, like STIs. So now that we've understand how different ways to have different ways in which we can have epidemic or spreading of disease, we also have to understand the epidemiological statistics relating to this disease. So first of all, we have to know the prevalence of the disease, which is basically the total existing cases with respect to the entire population. And this may or may not include asymptomatic infections. So all you have to do to fully understand the prevalence of disease is that you calculate the number of total disease you have in the population and you divide it by the whole population. And just if you want to put it in a percentage form, just multiply it by 100 and you have the percentage prevalence of a disease. For that, you can hear it in the news, like someone will be like, oh, the prevalence of influenza in the US for the year 2022 was about 0.5%. So that's basically how they come by this calculation. And then we have incidence of the case. So basically the number of cases over certain time periods. And this help you to establish the risks associated with the disease. So then you have the number of cases over the total number of susceptible people that and persons that you have. And then you calculate that and easily can be reported as the incidence of new Zika virus cases in Belize in 2016 was 55 per 1,000 people. So you just take the new cases and then divided by the number of susceptible person. Then we also have mobility. It's basically the number of symptomatic cases out of all cases in the population. Typically represent this as a percentage. So example is chlamydia. You can say chlamydia has a mobility of 25%, meaning only 25% of people are symptomatic with a chlamydia infection. The next one is mortality rate, basically the number of deaths in a population due to certain disease, and you often represent this as a percentage. So you can report it as there were 210 deaths caused by Ebola out of 400 cases in the United States in 2015, and then you can represent this as 53%. Then we also have the case fatality ratio, which is death out of those with symptomatic disease, and then they also are often represented as a percentage. A percentage. An example is you can say out of 5,694 symptomatic cases of malaria, there were 803 deaths, or 14% death. So try and work in the Nipah virus epidemiology. The assignment is on module, and then the part two will also include this video. Try and click on it and watch it, and also practice on your worksheet. I hope to see you guys on Friday so that we can work through some of the examples and uh, discuss some of the review for your exam 3. Hope to see you and have a good day. Bye. Please turn in your assignment if you haven't. The semester is almost at the end.